biotechnology is changing preclinical research. Our first speaker comes to us from the Ottawa Hospital Research Institute. Manoj Lalu is the Associate Scientist in Clinical Epidemiology and Regenerative Medicine. He's also an Assistant Professor in the Department of Anesthesiology and Pain Medicine, where his research on developing preclinical and translational biotherapeutics, namely cellular therapies for inflammatory disease and cancer.
So what I'll be talking about for the next 20 minutes or so is some disruptive paradigm shifts to try to reduce irreproducibility in preclinical bench work. So that's what a lot of the focus in my lab group is around. We have a dry lab and a wet lab as well too, and we're really focused around these issues of irreproducibility in science. So I'll actually begin with a little bit of a story first to begin with. So I suspect many of you are actually familiar with this particular study. It was published in Nature back in 2012. And it was by a couple of scientists, actually the vice president of research at that time at Amgen, so one of the largest pharmaceutical companies in the world. And in this particular paper in Nature, they described their efforts to try to reproduce 53 papers that were published in high-impact journals like Nature, Science, and the like. And they described their efforts to try to actually reproduce the findings of these 53 papers. So I'll just put this out as a question to everyone. So how many of those 53 papers do you think they actually reproduced? Was it 53, so was it 48, 24, 12, 6, or 0? So who thinks it was, just by a show of hands here, who thinks it was 48? Who thinks it was 24? Who thinks it was 12? Who thinks it was 6? And who thinks it was 0? Okay, so the answer was actually 6. So 6 of the 53 findings. And this is Amgen, so this is, and there was a vice president of research actually conducting all of this as well too. So literally millions of dollars were poured in to try to reproduce these findings, and they were only able to reproduce 6 of them. More recently, there's been another project called the Cancer Reproducibility Project. It was funded by the Center for Open Science. So if you just Google the Cancer Reproducibility Project, there's a whole series of papers that are published in eLife, and they've been funded to look at 18 again, major findings that they're trying to reproduce. They've completed 14 of those studies, a little bit better than this in that they were able to reproduce 5, but that means over half of these major findings they could not reproduce major aspects of those studies. So obviously a very big problem, and something that's been highlighted in a lot of different editorials in the last few years as well here. So I've just put a little collage here together of all these different editorials in different journals and even the lay press as well too, talking about irreproducibility in basic science and why that's a major issue. It's even been highlighted in major clinical journals like the British Medical Journal. So it's a journal, top-tier medical journal, largely clinical. You don't find any preclinical work in there, but they're even highlighting it in a cover article at the end of last year saying, hey, this is a big problem and it's actually not allowing us to best inform the health of our patients because of some of the irreproducibility problems within preclinical science. So my group and many others are looking at these aspects of irreproducibility and how we might be able to actually disrupt this particular problem. So I think there's 3 major aspects that lead into this irreproducibility. One of the major issues I haven't even put up here is just scientific fraud. So basically data mismanagement, and I think that's a major aspect of a lot of the irreproducibility that we see. However, that's not going to be the focus of the next few minutes. What I'll be talking about instead are issues that I think people in this room can help us deal with. Number 1 is poor reporting, and number 2 is what we call risk of bias. So what do I mean by that? I mean things like randomization and blinding and sample size calculations. These are things that are commonly done in clinical studies, and even these are clinical studies I'm talking about even in sort of lower tier journals. However, these are things that we don't routinely do in the lab, and perhaps we should be thinking about doing that more often. So I'll provide some evidence around that as well too. And it's not just me and a few other groups around the world, it's actually major funders that are talking about this as well. So this is from the NIH. It was published in 2014. Francis Collins, the director of the NIH at that time, wrote this particular editorial. And it was actually in direct response to that first slide I showed you from that Amgen paper. So they saw that there was a lot of buzz around that particular paper in Nature, and this was the NIH's direct response to that, saying, hey, we're going to try to do something about this. So within that editorial, they highlighted two issues that I'll be, again, talking about today. The first little box there talks about factors that contribute to irreproducibility include poor training of researchers in experimental design. And then the second factor they highlighted is that publications do not report basic elements of experimental design. Crucial experimental design elements that are all too frequently ignored include blinding, randomization, replication, sample size calculation, and the effect of sex differences. So I myself was primarily trained as a basic scientist before I went into clinical medicine, and I still have a wet lab, like I mentioned. And again, these were issues that I certainly never learned about during my PhD, formally at least, or one of the two postdocs that I did as well after that. So again, these are things that 10 years ago, I don't think there was much around it. There is now beginning or developing a bit of a core of resources that are actually attacking these issues and hopefully highlighting the importance of them. So solution number one, again, I said I was going to talk about, is improving our preclinical reporting. And what do I mean by that? The best example, and I just love this, is from Anesthesiology, so it's a top-tier clinical journal, actually, where they do actually publish some animal work as well, too. And they're talking about reporting and what they're going to do in reporting. And the example they use is if a clinical study was actually reported in the same way or conducted in the same way that we often do our exploratory work in the lab. So what they said was, and this again is hypothetical, they're talking about this hypothetical clinical study that if it was conducted the way we typically do exploratory research in a lab. The investigators had not performed these tests before, so they decided to give placebo for the first 20 patients. The results were consistent with other studies of recovery from sedation, so gave the active drug to the next 20 patients. They examined the results and noted that one of the outcomes, speed of finger tracking, showed a large but variable drug effect in the anticipated direction, but only at 30 minutes after surgery. Measurements were actually taken at 15, 30, 45, and 60 minutes after surgery. They used several statistical tests to compare the groups for this outcome, and the one that was closest to statistical significance showed a p is equal to 0.09 after they excluded one patient receiving active study drug who had a longer time than the others. Based on these promising results, the investigators enrolled 2 more patients per group. This resulted in a p is equal to 0.06, so they enrolled 1 more patient per group and observed a statistically significant effect of p is equal to 0.048. They rejected the null hypothesis and stopped the study. So this seems sort of ridiculous when you think about it in a sort of patient study, but this is typically what happens in exploratory research, and I think there's certainly a place for it. Again, I have a lab. I've worked in labs in many years for my life as well, too. I realize how things happen, but the issue here is that if you're doing this, you should probably report it as well, too. So that way, people can make a transparent assessment of how much confidence they can have in your results or what further work needs to be done to confirm that particular finding that you had. So again, the NIH has looked at this issue, and this is what they came out with. It was the principles and guidelines for reporting in preclinical research. So if you just Google NIH and preclinical reporting guidelines, you'll find this particular website. And they talk about 7 core elements, and I'm just providing a high-level overview today, so we're not going to go into all these in detail. But you can see the things I've already mentioned are there: randomization, blinding, sample size estimation. And they're not saying you have to do this. They're not saying that at all. They're saying just simply say if you've done this or not. And then people who are reading your report can make an accurate assessment as to how much confidence they can have in your results. And again, what further work needs to be done to build on whatever your finding has been. So how good are we at actually reporting these elements right now? And our group has looked at this over the last year, and these are the 7 different domains that I just mentioned there for the NIH reporting guidelines, and these are on the y-axis the percent of papers reporting on this. We looked at 2 groups of papers, those that were reporting on oncolytic virus findings. This is all preclinical, and there's 200 of those, and then preclinical anesthesia papers. Again, I'm an anesthesiologist, so that's where that came from. But there's 600 papers there, all preclinical. So close to 1,000 papers there, and you can see the percentage of reporting is actually very low between those 2 groups. Red is the anesthesia, blue is are the OV papers. So outside of statistics, Pretty much everything else is under 50%, and it drops down to almost zero when you're talking about standards. By that, they mean, did you actually use a reporting guideline? And the reason that's important is in the clinical world, you can't actually publish in even a mid-tier journal without attaching an appendix saying, hey, I've done an RCT, these are my consort guidelines. So consort is just a reporting guideline for randomized clinical trials. Or this is my observational study, and on my appendix here, or my strobe, is my strobe checklist saying I've actually reported on all the things that are required for an observational study. So preclinically, there's the NIH guidelines. There's also another set of guidelines called the ARRIVE guidelines, which was just updated this, I think 2 months ago now. These are reporting guidelines for preclinical work, and now even top-tier journals. So Nature Medicine, for instance, if you submit there is a checklist that you actually have to fill out as well, saying, did you do a sample size calculation? Did you randomize? Did you blind? And again, if you didn't, that's fine. It's not a penalty on your paper at all. They're just wanted to have a transparent reporting of these issues so that you can actually assess the confidence of those findings. So things are changing in terms of reporting, and if top-tier journals are doing that now, you can bet in the next 5 years that more and more journals are going to pick up this and be demanding that when you submit your paper from whatever your findings were in the lab, that you actually fill out a checklist as to what you're reporting in your paper. So the next piece I'll be talking about is education around preclinical rigor. And I'm sure many of you here have maybe not really thought around these issues of randomization and other methods, that might reduce bias in the lab. And you might think, well, it's not that important because I'm using inbred mice, they're all the same, so who cares? Why should I randomize? Why should I blind? But we know from really landmark paper, this is actually 10 years old now, that there's a mountain of evidence that's accumulating, demonstrating that things like randomization, blinding, and sample size calculation, in other words, rigor at the bench, really do make a difference. So in this particular paper, it's a really interesting story. They actually did what's called a systematic review. I'll be explaining that in actually a later slide. But they basically looked at every single study preclinically where they'd given this particular compound, NYX059. It's an antioxidant agent, and it was shown preclinically to have a great deal of promise. However, then they went on to conduct a trial. It was a multicenter phase 3 trial called the SAINT-2 trial. And there were several thousands of patients enrolled in this multinational trial. It completely failed. And the clinical side of the stroke world said, hey, there was so promising in the preclinical, all these preclinical findings, NYX059, why did it fail? How much confidence can we have at all in any of these preclinical findings if this drug that had this large effect showed no effect in humans? So this group from Scotland, Malcolm McLeod's group, they actually performed a systematic review to identify, again, every single preclinical study where they'd given NYX059. And I'll just show you, I'll orient you to the axis here, first of all. So this is stroke, so it's reduction in infarct volume is the y-axis there. And then across the bottom here, they actually, first what I'm going to do is actually orient you to, if I can figure out this cursor here, is this big gray bar here. So these are, if you took all the studies together, all the preclinical studies of NYX059, that is the effect with the constant interval of what the reduction in infarct volume would be, so 50%. And then what they did is they actually split these studies into those that had randomized versus not randomized in the left side, and those that had blinded versus not blinded. You can see there is a clear difference in the effect size between those that had randomized and those that had blinded. Again, this is one of the landmark papers demonstrating that randomization and blinding actually make a difference in the lab. Subsequent to this, there's been many, many papers demonstrating similar effects as well, too. So things like randomization and blinding and actually developing your protocol ahead of time might make a difference. So this is from Nature just a couple of weeks ago, and they're talking about animal registries. So just like we have registries for clinical trials, they want to start setting up registries for what we do in the lab as well too. So before you conduct a study, you go to this place, you would register your study, say this is what I'm going to do, and then do it. And then your final report, you can actually refer back to that protocol and say, hey, this is what we plan to do, this is where we deviated from the protocol, these are our findings, basically. The problem with that, and I was actually interviewed for this particular paper, the problem with this aspect is I don't think people understand what the point of all these things are. What is the point of blinding? How do you do it? What is the point of randomization? How do you do it? What is the point of a registry? And what should you actually fill out in that? What I was quoted there was that still the availability of registry is just one piece of the puzzle. Many researchers do not understand why techniques to reduce bias are necessary or how they should be implemented. This means that even if they do register a study, they may do so inaccurately. Thus, registries must be combined with educational resources. So luckily within Canada, Biotherapeutics and now SCN have actually really stepped up to the plate and said, hey, we're going to try to educate our researchers around these issues. So through Biotherapeutics over the last couple of years, once a year we've brought together groups to actually teach them how to randomize, how to blind, how to do a sample size calculation before you actually proceed with an animal study. So this is just the front page for the workshop that we did last year in Banff with Biotherapeutics, and this is just a picture of there's about 30 or so people that were in the room, senior investigators, some people from CROs were there as well too, as well as trainees. And we just basically covered all the 7 elements that were in the NIH reporting guidelines, told people how to actually implement them in the lab. In that picture is actually Carolina Ilkow, who's one of the speakers. She's one of the superstar researchers in Biotherapeutics, and she's published a couple papers in Nature Medicine recently as well too. So she's telling the crowd there why it's important to blind and how she actually does it in her lab. And then just a plug for February this year, SCN is actually sponsoring this for this year. So if you're interested in this sort of aspects, we're going to be running this workshop again in the spring. In February. So next solution is preclinical systematic reviews. Again, I did mention that in a previous slide because that was where that NYX 059 data, came from. So what's a systematic review? I'm sure almost everyone in this room has written or contributed to a narrative review. So a narrative review, you usually just pick out different things you're interested in, you talk about them, you might ignore things that sort of go against your hypothesis, and that's fine for a narrative review. But a systematic review is actually very, very different. A systematic review is a review in which there's a comprehensive search for relevant studies on a specific topic, and those identified are then appraised and synthesized according to a predetermined and explicit method. So this is a definition of clinical systematic reviews done by some of the godfathers of clinical systematic reviews. The whole methodology is actually very old. It came from psychology originally, was adapted about 20 or so years ago by the clinical world, and in the last 5 to 10 years has now been adopted by the preclinical world as well too. So the reason you would want to do this is that you get an unbiased and up-to-date summary of the efficacy or effectiveness of a therapy preclinically. And I'll show you our example where we actually did that prior to conducting a first-in-human study. And this, again, is a very novel sort of technique. There's an explicit method, there's best practices around it. You might have heard of the Cochrane collaboration or Cochrane review. This is the sort of premier organization clinically that conducts systematic reviews in that domain. Preclinically, there's a few different organizations now promoting it. And this, again, is very much an emerging technology. So this is just the number of systematic reviews preclinically that have been published from 2005 onwards. You see the scale starts at 5 there approximately, 14 years ago. 2012, we have 30. And then my group has actually just over this past summer looked at how many have been published more recently. And you can see the scale's totally changed now. In the last year, we've had over 160 preclinical systematic reviews being published. So this is very much an emerging area. And I'll just have 2 slides here very briefly on just as an example of why you might want to think about doing this. So this is for a systematic review of mesenchymal stem cell therapy for sepsis prior to a trial that's actually been completed now that the SCN had actually funded. So what we did was we looked at all the preclinical studies prior to actually even designing the protocol for this particular clinical study. We gathered all the different preclinical studies, so in vivo animal models of sepsis where they had given MSC therapy. And we looked at the outcome of mortality. These are the 19 or so studies that we had found that had looked at this. This is called a forest plot. I'll just orient you very briefly to what this actually is about. So you have individual studies here on each line. You have the point estimate for mortality as a dot, and then a confidence interval as a line that goes through it. If it's on the left side of one, it favors MSC therapy to reduce death. If it's on the right side of one, it favors control animals over the MSC therapy. And then what we did on the very bottom here is a meta-analysis, so it's just a statistical method to combine all these studies together, and you can see that there's a pooled estimate there clearly demonstrating that MSC therapy reduces death in these animal models. Overall, what you can see here, again, it's just think about reproducibility, is that we're able to demonstrate that this is a finding that has been reproduced over several different independent laboratories. So finally, solution number 4 to irreproducibility is multicenter preclinical studies. So what are multicenter studies? Well, these are when you have multiple independent laboratories performing the same experiment using a shared protocol. This is the gold standard in clinical research. And why is that? Because it rigorously evaluates reproducibility between centers that are performing this clinical study. So my center, the TOH, and your center at the U of T can actually produce the same results with a similar sort of patients in a clinical trial protocol. And again, gold standard, and in fact, there's been multiple meta-research studies saying that single-center trials show larger treatment effects than multi-center trials. Why we should be wary of single-center trials, another one, and then a similar thing from the BMJ, saying that really we should be paying attention to multi-center trial data over single-center trial data. So then I ask the crowd here, and the people I work with, why do we place so much emphasis and confidence in single laboratory findings? Would it not be better to actually try to do multi-center, multi-laboratory studies and see if we can actually produce the same results? Again, very much an emerging area. This is from Science Translational Medicine in 2015, where they did exactly that. This is a stroke model where they gave an anti-CD49 antibody to treat stroke. Very, very promising findings in single-center studies. They gave this drug to 5 different centers and basically blinded, randomized, so they didn't know what they were giving. They didn't know if it was control or treatment animal. Everything was analyzed in a blinded manner as well, too. And what you saw overall is that, so the white is control, the shaded is our treatment groups there, and there was no effect overall. So exactly what we see clinically. So this is the exact same sort of findings you see clinically, single-center clinical studies, huge promising findings, move it to a multi-center laboratory, multi-center clinical trial, and these findings fall apart. So we've actually done a systematic review in this area, try to identify every single preclinical multi-center laboratory study that was conducted. There's only 12 that have been conducted ever. The first was 1985, and the last few have been published in the last couple of years. So of these 12 that we were able to find, 6 were unable to replicate the results of the single-center findings that led directly into this multi-center laboratory study. That was the last slide. So, the 4 of the studies were actually able to replicate those single-center findings. A couple of studies have now proceeded to first-in-human or early-phase clinical trials. So again, a very much an emerging area. There's going to be a tidal wave of these coming through. I was on a grant review panel in Europe where a couple of different federal funders are now specifically funding multi-center preclinical studies. Unfortunately, in Canada, it doesn't exist yet. Hopefully it will. But again, very much an emerging method that you'll be seeing more of. And then just a final plug for some work that the OIRM has funded for our group, where we're actually going to be conducting Canada's first multi-center preclinical study, where we have 3 different labs, 2 from Ottawa, 1 from U of T Haibo-Zhang, where we're going to be giving MSC therapy, so stem cell therapy, to a lung injury model. Our first paper out of this should be coming out, or submitted at least soon. And again, very much an emerging area, and hopefully you'll be seeing more of this in Canada. So last slide is just my acknowledgements here. I'm on Twitter. I do tweet about reproducibility every once in a while too, as well as anesthesia. But that's my handle there, and those are people that I collaborate with, and that's my research group in the picture there. So thank you very much for your time.